The overall goal of the following experiment is to construct gene-targeting vectors using subcloning plus insertion. This is achieved by transforming the back containing E. coli with a recombineering plasmid. The second step involves generating asymmetric phosphorothioated insertion cassettes and subcloning plasmids by polymerase chain reaction. Following the addition of arabinose, the GBA-A recombineering proteins are expressed, and the terminal modified insertion cassette and subcloning plasmid are electroporated. The desired DNA sequence is simultaneously captured from the back and modified with the insertion cassette into the subcloning plasmid.
The main advantage of this technique over existing methods like standard recombineering is that gene-targeting vector construction can be performed in a single reaction.
To begin, design oligos for the insertion cassette and the subcloning plasmid. Design each oligo such that it contains 180 base pair homology arms flanking the genomic target site, and 20 base pairs of specific priming sequence for the insertion cassette or subcloning plasmid. Next, determine the orientation of the genomic DNA insert of the back clone using a web-based tool like CloneDB. Establish the direction of replication from ORI-S by checking the map of the back plasmid backbone used in the back library construction. Add 2 terminal phosphorothioate bonds to the 5' end of the oligo that is opposite the direction of replication on the back clone. Next, add a 5' phosphate modification to the reverse oligo.